Hi, my name is Ashwinder and today we'll be learning about E. coli. Where does the name Escherichia coli come from? The genus Escherichia is named after a German pediatrician, Theodor Escherick, who made contributions to the understanding of intestinal bacteria. So Escherichia honors Dr. Escherick. Coli, the second part of the name, signifies the species, and it's derived from the Latin word colon, indicating that the bacteria is commonly found in the colon of humans and other animals. What is the gram stain result? E. coli 0157 is a specific serotype of the bacterium Escherichia coli, and its gram stain result is gram-negative. Gram staining is a laboratory technique used to categorize bacterial species into two broad groups, gram-negative and gram-positive, and it's based on the characteristics of their cell walls. Gram-negative bacteria like E. coli have cell walls that contain a thin layer of peptidoglycan surrounded by an outer membrane. This thin layer of peptidoglycan does not retain the crystal violet stain, which is used in the gram staining process. And as a result of that, gram-negative bacteria usually appear pink or red under a microscope after staining. The use of a microscope. When examining E. coli under a microscope, it's important to note that E. coli bacteria are typically very small and not easily visible with the naked eye. Microscopy allows for the visualization of individual bacterial cells and their characteristics. And here's how you would use a microscope to study E. coli. You prepare the sample on a glass slide, taking a sterile loop and a loop full of the bacteria and smearing it in circular motions in the middle of the slide. And then you stain the sample using crystal violet, and then you set up the microscope by using the coarse knob. Then you focus the microscope using fine focus and the iris lens. You can change the objective lenses as you go on, and then you observe. Structures and functions that correspond with the gram stain results. The first one is cell wall. It contains the peptidoglycan layer, and the peptidoglycan layer is thin, and it's located in the periplasmic space between the inner and outer membranes. It consists of a single layer of peptidoglycan. The peptidoglycan layer provides structural support to the cell and helps maintain the cell shape. The outer membrane is a unique feature to a gram-negative bacteria. It consists of lipopolysaccharides, proteins, and phospholipids. The outer membrane is asymmetric, with the lipopolysaccharide layer facing the external environment and the inner phospholipid layer facing the periplasm. The outer membrane acts as a barrier to many substances, including antibiotics and detergents. It also contains porin proteins that allow the passage of specific molecules, like ions and nutrients. The inner membrane. It's also known as the cytoplasmic membrane. It's a phospholipid bilayer that surrounds the cytoplasm of the bacterial cell. It is located beneath the peptidoglycan layer. The inner membrane is essential for maintaining the integrity of the cell and regulating the passage of molecules in and out of the cell. The cytoplasm is a semi-fluid interior of the cell where many cellular processes take place. It contains the DNA, ribosomes, and various proteins. The cytoplasm is the site of numerous biochemical reactions, including metabolism, protein synthesis, and DNA replication. What is responsible for Anna's diarrhea? E. coli is a specific strain of the bacterium that is associated with causing gastrointestinal illnesses, including diarrhea. This strain of E. coli is particularly known for its ability to produce a toxin called shikatoxin, which can lead to a condition known as shikatoxin-producing E. coli infection. Some of the symptoms are diarrhea, abdominal pain, nausea and vomiting, fever, and fatigue, and loss of appetite. What caused Anna's slimy feces? Pathogenic E. coli strains can cause inflammation in the intestines. This inflammation can disrupt the normal processes of the gastrointestinal tract and lead to changes in stool consistency. The lining of the intestines produce mucus as a protective barrier against harmful substances. When the intestines are irritated or inflamed due to an E. coli infection, the body may produce more mucus in an attempt to protect itself. And this excess mucus can mix with stool, making it slimy and mucousy in appearance. E. coli infections can interfere with the absorption of water and nutrients in the intestines. When the intestines are not functioning properly, it can result in diarrhea, which can lead to loose or slimy stools. Antibiotics. Many cases of E. coli infection resolve on their own with proper hydration and supportive care. The body's immune system can often fight off the infection without the need of antibiotics. Overuse of antibiotics can contribute to the development of antibiotic-resistant bacteria, which is a global health concern. It is important to use antibiotics judiciously and only when necessary to prevent the development of antibiotic resistance. In Anna's case, if she is recovering well, staying hydrated, and not experiencing severe symptoms or complication, it is typically best to allow the infection to run its course without antibiotics. Thank you.